Good afternoon, everybody, and welcome of my talk. Thank you for the kind introduction, and it's an honor for me to, and I'm thankful for organizers for giving me the opportunity to give a talk here. And I will start my presentation with a short, simplified geographic scheme of my travel to Glycobiology World Congress. So I did my master equivalent in Belarus. This is a country in Eastern Europe. And after that, I moved to northwest of Germany, to Münster, where I joined a group of Professor Johannes Mütting. So I did my PhD there, and now I'm doing a postdoc. And currently I'm here in Philadelphia to give an overview of what our working group is doing. So first of all, I will start with a short introduction, and then I will go to shiga toxin glycosphingolipid receptors of human endothelial cells. After this, I will talk about lipid raft association of shiga toxin glycosphingolipid receptors. And finally, we'll talk about shiga toxin-mediated damage of human endothelium and blood-brain barrier, and also we'll give some perspectives. So on this slide, you can see well-known microorganism, Escherichia coli, which is a normal commensal of human intestine. However, due to acquisition of different virulence factors, it also hides tremendous pathogenic potential. And so it has nothing to do with bioterrorism or whatever, so this is what happens naturally due to evolutionary means. And there are different pathotypes of Escherichia coli that do exist, but here I will focus on enterohemorrhagic Escherichia coli as, or EHEC, because EHEC is relevant to our research. And outbreaks of enterohemorrhagic Escherichia coli, so they happen in worldwide. And so often EHEC appears as a hero in the news, of course in negative connotation. And here I will just shortly summarize some EHEC outbreaks that happened worldwide. And actually the first one that was registered in 1982 actually happened in the USA. And it was caused by EHEC strain O157H7, which is now considered to be more closely associated with the development of the disease. So these outbreaks they happen worldwide. However, they also can be caused by different other uncommon EHEC strains. For example, there was a very large EHEC outbreak in Germany in 2011, which was caused by a typical EHEC strain O104H4, which actually combined virulence factors of both EHEC and enteroaggregative Escherichia coli, and there were like more than 3,000 cases of infection and even 54 deaths. So EHEC, they cause a wide range of different symptoms such as watery diarrhea, bloody diarrhea, and the worst case is hemolytic uremic syndrome. And here we can see the development, the scheme of the development of infection caused by EHEC. It starts with occasional ingestion of the pathogen. Then in a couple of days, there are first symptoms such as diarrhea, abdominal pain, fever, and vomiting. In a couple of days, bloody diarrhea occurs in 90% of cases. And then approximately in a week, in 85% of cases, there is spontaneous resolution of the disease. However, in 15% of cases, hemolytic uremic syndrome occurs, and this is the worst case scenario. So it's characterized by hemolytic anemia, thrombocytopenia, and acute renal failure, and it leads to, in lots of cases, it leads to the death of the patients. So what makes EHEC so infectious? So there were several virulence factors identified. For example, intimin, which is responsible for formation of pedestal-like structures on epithelial cells, EHEC hemolysin, and there are some other, also not identified so far, virulence factors, but the main factor which is involved in all severe cases of the disease, this is shiga toxin. And shiga toxins, these are ribosome activating proteins. They contain a catalytically active A subunit, which has rRNA and glycosidase activity and inhibits protein biosynthesis. And there are also 5 identical B subunits, which are responsible to binding to glycosphingolipid receptors. And there are 2 main types of shiga toxins. These are shiga toxin 1 and shiga toxin 2, and also a set of subtypes, and about this I will talk later. So shiga toxins, they bind to glycosphingolipids, and here is like a short scheme of biosynthesis of shiga toxin glycosphingolipid receptors. It starts with the formation of glucosylceramide in the endoplasmatic reticulum, which is then stepwise glycosylated in Golgi apparatus. So lactosylceramide is formed by the activity of beta-1,4-galactosyltransferases by adding terminal galactose, and lactosylceramide is further elongated by the activity of alpha-1,4-galactosyltransferase, yielding globo-triazylceramide or GB3-ser, and GB3-ser can be further elongated by the activity of beta-1,3 and acetyl-galactosamинyltransferase, forming GB4-ser or globo-tetrazylceramide, and in some cases, GB4-ser can be further elongated by the activity of alpha-1,3 and acetyl-galactosamинyltransferase, forming Forssman glycosphingolipid. And these last three glycosphingolipids, they are relevance of interaction with shiga toxin since, and this will be of the focus on the next slides. So if to have a closer look on glycosphingolipids on the example of globo-triazylceramide, so we can see that it has oligosaccharide chain, which is responsible for recognition of shiga toxin, and it has hydrophobic ceramide moiety with which it's anchored in the plasma membrane. And in ceramide moiety, we can distinguish sphingosine, which appears to be constant in most human cells with 18 carbon atoms and monounsaturated, but fatty acid is quite variable from C16 to C24. Here it's only shown with C16. Now I would like to particularly talk about glycosphingolipid receptors of human endothelial cells, and so on the next slides I will present our studies on glycosphingolipids isolated from endothelial cells of brain and kidney, because brain and kidney, these are the main organs that are targeted by shiga toxins. Here you can see the results of detection of glycosphingolipid receptors to shiga toxin in human brain microvascular endothelial cells and glomerular microvascular endothelial cells of kidney. If to make a long story short, so glycosphingolipids isolated from cells were separated on thin layer chromatography plate, and then specific antibodies were used for different types of glycosphingolipids. And what we can see here that both cell lines from brain and kidney, they do express globo-triazylceramide, which appears here as a double band because of fatty acid heterogeneity. And also they express globo-tetrazylceramide, which was also shown by overlay assays. In order to obtain structural information, mass spectrometry experiments were performed, and here I exemplarily showed the diversity of globo-triazylceramide in glomerular microvascular endothelial cells. And what we can see that we have GB3-ser with short chain fatty acid, which is in the lower band, and in the upper band we can find globo-triazylceramide with C22, C24 saturated and monounsaturated. Later we confirm the structures with MSMS experiments, but here I will not show it because of lack of time. And now I would like to focus on different binding of STX1A, 2A, and XTX2E to glycosphingolipid receptors. And for this case, we applied equimolar concentration of globo-triazylceramide, globo-tetrazyle, and Forssman, and performed binding experiments using supernatants from Escherichia coli that were containing particular types of shiga toxins. And what we can see here that in case of STX1A, it recognizes nearly equally GB3-ser and GB4-ser, but not Forssman. And similar pattern is for STX2A. It recognizes GB3-ser and to a little bit to less extent of GB4-ser and no recognition of Forssman. However, if we use STX2E, we can see that it binds both to GB3-ser, GB4-ser, and Forssman, and it holds true for isolates that we obtained from both human strains and from porcine. And what we know that STX2E, it's responsible for peak edema disease, and only this STX2E recognizes Forssman glycosphingolipid. So summarizing this, so there are three main glycosphingolipids that are involved in shiga toxin interaction with shiga toxin. This is globo-triazylceramide and STX1A, STX2A, and STX2E. They all recognize GB3 as well as GB4, but concerning fourth small glycosphingolipid, only STX2E recognizes it. And now I would like to switch to lipid raft association of shiga toxin glycosphingolipid receptors. And so lipid rafts are microdomains of the plasma membrane that are enriched in cholesterol, GPI-anchored proteins, and glycosphingolipids. And they're responsible for signal transduction, but at the same time, these are platforms for entry of pathogens and their toxins. And what was shown by many investigators that lipid raft association of shiga toxin glycosphingolipid receptors, this is a prerequisite for retrograde transportation of the toxin to intracellular targets. And the key method of investigating of lipid rafts, this is preparation of so-called detergent-resistant membranes. And to explain it in a simple way, so membranes isolated from cells are treated with a detergent, in our case it's Triton X100. And then detergent intercalates in non-raft fraction of the membrane, and then micelles are formed. However, raft fraction of the membrane remains intact, and when we, after treatment with detergent, we perform ultracentrifugation of the membranes in the sucrose in density gradient, and then we can separate these different parts of the membrane. And we can see that detergent-resistant membranes that correspond to lipid rafts, they're floating in the upper first three fractions. And the rest of the fractions, they correspond to non-lipid rafts. And so we collect the fractions from top to bottom, and then we perform subsequent analysis. And here I will exemplarily show the results of analysis of detergent-resistant membranes of human brain, microvascular endothelial cells, and glomerular microvascular endothelial cells in HBMX. So we can see that DRMs, they are enriched in cholesterol and glycosphingolipid receptors GB3 ser and GB4 ser, and non-lipid raft fraction of the membrane, they have only fade amounts. And this is what we normally find in many types of cells. But however, we found quite unexpected results for endothelial cells that we isolated from kidney. In this case, we found that mainly glycosphingolipid receptors are in non-DRM fractions as well as cholesterol. We still cannot really explain these results. And now I will go to the topic of STX-mediated damage of human endothelium, and this is a work that have been performed in our institute by Dr. Andreas Bauens, and he was growing endothelial cells on microcarriers, and using scanning electron microscopy, he was visualizing different effects of STX1A and 2A. So on this slide, you can see the scanning electron micrographs of intact endothelial cells, and we can see that they have a typical cobblestone pattern, and there is strict contact inhibition of the cells. However, if the cells are treated with XTX1A, we can see the irregular shape of cells, we can see membrane blabbing, gaps between cells, and in general, severe monolayer damage. And these are signs of both necrosis and apoptosis. However, in case of STX2A, it was shown that STX2A induces mainly apoptosis. So there are only some membrane blabbing, but no plasma membrane lesions or cell detachment. And if to talk about susceptibility of these two different cell lines from brain and kidney to shiga toxin 1A and 2E, we can see that they react in quite dose-dependent manner. However, what we see that endothelial cells from brain, they are less sensitive to both shiga toxin 1A and 2A compared to endothelial cells from kidney, and it's quite surprising because the common idea is that lipid raft association of glycosphingolipid receptors, this is a prerequisite for toxin susceptibility. However, we find that in endothelial cells from kidney, glycosphingolipid receptors are not located in lipid rafts. However, they are more sensitive than endothelial cells from brain. So it was estimated that endothelial cells from kidney, they are about 1,000 times more sensitive to STX1A and 2A than endothelial cells from brain. And finally, I will just mention one of our projects, which is concerned with blood-brain barrier. And on this slide, you can see the circulation of the brain. And blood-brain barrier comprises of endothelial cells, which form the lining of the blood vessels, and changes of blood-brain barrier permeability can lead for the penetration of toxins and different other substances can make it more easily. We have been studying the loss of blood-brain barrier integrity upon exposure to shiga toxin 2E containing supernatants from bacteria. And what we know that shiga toxin 2E is responsible for edema disease and PICS, and this is a disease which is characterized by disruption of blood-brain barrier and PICS, and the common signs is hemorrhagic colitis and dysfunction of the brain, and so a lot of animals are dead. So to study blood-brain barrier, in this case, so we used primary porcine brain endothelial cells. It was isolated from PICS, and then so these cells were grown on an in vitro system on filters, and so they were separated in apical and basolateral side, which correspond to blood and brain. And we were measuring trans-endothelial electric resistance of the cells, and what we can see in controls when cells were just treated with supernatants from bacteria that were not containing any toxins, we can see that there was slight decrease in trans-endothelial resistance. However, it was stable during 24 hours. However, if we were applying STX2E, we saw the rapid breakdown of blood-brain barrier. So like after 2.53 hours, there was like 50% decrease, and then after 8 hours, it was completely not functional. So at the final stage of my presentation, I would like to give some perspectives. So there could be investigation the functional role of different lipoforms of shiga toxin glycosphingolipid receptors. So we see some receptors heterogeneity in fatty acid, but we don't really know right now the functional role of it in the cells. The next could be unraveling the role of plasma membrane microenvironment, so-called lipid raft on STX binding and internalization, and defining exact molecular mechanism by which shiga toxins interact with cellular targets. And this in the future can help in developing new strategies aimed at preventing toxin binding and internalization, and new complex specific therapeutics and protective measures for EHEC-mediated diseases. So at the end of the presentation, I would like to acknowledge our group leader, Professor Dr. Johannes Muting on the left side, and director of our institute, Professor Dr. Helge Karch, also Dr. Andreas Bauens, and all the members of the group. And thank you for your attention.
Very good. Excellent presentation. Any questions for the speaker?
What is the stability of the shiga toxin? First one. Second question is, what is the recognition size of the shiga toxin? Two questions.
But concerning the first question, what do you mean by ability?
Stability. How stable?
So you mean in vitro or in vivo?
Both.
So concerning the stability of in vivo, so what I could say that like a very small amount of toxin is enough to intoxicate the cells. And concerning the stability of in vitro, so there was like a work of Professor Maurizio Brigotti from Italy, and he is working on the transportation of shiga toxin in the bloodstream. And what he was shown that, like, for example, repeated melting and thawing circles of shiga toxin might lead to inefficient binding of the toxin. And also, different groups, they get contradictory results because they use different purification schemes of shiga toxin, and like the more complicated the scheme for purification, there is the more probability that toxin, for example, might still exert its cytotoxic activity, but it can be not. It may not be able to bind. And your second question?
Yeah. Second is, what is the recognition size and structure? That this shiga toxin recognize what kind of structure, and then the other is they recognize a critical structure or not.
So epitope structures is it recognizes alpha 1, 4, beta 4. So these two terminal sugars, and so globotriazylceramide is the main receptors. The main receptor. However, if there are some other terminal sugars added, as in the case of globotetrazylceramide, so shiga toxin can still interact with globotetrazylceramide. However, we don't know really if shiga toxin binds to globotetrazylceramide, whether it's internalized. And concerning, for example, even more prolonged structure of Forssman, which has like additional GalNAc at the very end. So what we showed, like what I showed in the other slide, that for example, some subtypes of shiga toxin, like shiga toxin 1A and 2A, they are not able to recognize this structure anymore when there are two terminal GalNAcs added. But for example, shiga toxin 2E can do this.
GalNAc also involved in shiga toxin. GalNAc and acetylglycosamine. In the toxin receptor.
So the main epitope is these two galactoses, and the most important, this terminal with alpha 4. But if there are some other sugars added, so shiga toxin can still recognize the structures, but this is not the target. The target is mainly this terminal disaccharide, which we can find on globotriazylceramide.
Because in my knowledge is shiga toxin recognize galactose alpha 1,4 galactose, and then
yeah, galactose alpha 1,4. So this is what is. That's why globotriazylceramide is the main receptor for shiga toxin.
One more question.
I thought that was really interesting, the selectivity, from the different shiga toxins, these 2E, and because I didn't realize that one of them actually bound Forssman. I think that's very fascinating. I mean, is that does shiga toxin use that in different, like Forssman is not a human antigen, it's usually in animals, and so is that how it infects certain animals? Certain ones will infect different animals, like shiga toxin 2E or whatever, like this. Yeah, this I thought that was a very interesting.
Shiga toxin 2E is not relevant for human diseases. So because like Forssman is not presented on the surface of human cells, but it has relevance for animals.
And so structurally, does anyone know why it will actually bind to Forssman? Putting two other GalNAcs on there.
This is not really known, but there are some speculations that binding. So this is the surface of B pentamer, and there are like 3 binding sites on each B subunit, and there are some speculations that these binding sites can be somewhat different, and then they can influence the ability of shiga toxin to bind. But this is what we don't know for sure, but there are some talks about this.
Thank you very much for the questions. Any more? We can move on. Thank you very much for the excellent presentation.
Thank you.
Very good.